Thank you, Dean. It's a real pleasure today to give you a brief overview of the ongoing clinical trials in Alzheimer's disease, and I'll focus on the ones that we have here at UBC. Now, as the official timekeeper of today's forum, I've given myself -5 minutes, so it's going to be a bit of a challenge, but we'll try. So first of all, just to get everyone on the same page, this may be obvious to most of you, but what is a clinical trial? And a clinical trial simply is a setting where we test new drugs in patients with the relevant disease, in this case, that being Alzheimer's disease. But before a drug reaches a clinical trial, it needs to go through many steps. And Dr. Wellington told you how we have to develop this in the laboratory before it reaches a clinical trial. And even before most of the trials that you may see, it's already been in several earlier trials before it reaches that stage. But before you have a trial, you have to know what you're going to treat. And I'm going to highlight 2 aspects of brain dysfunction that we try to treat in Alzheimer's disease. And you've probably seen this before. So this is an image on the left of an amyloid plaque. And it turns out that in the brains of someone with Alzheimer's disease, amyloid plaques build up. And we think of them as sort of gunk that builds up in the brain that we try to get rid of. On the right-hand side, you see something called the neurofibrillary tangle, which consists of tau. Those are the 2 main pathologies that build up in the brain of someone with Alzheimer's disease, and there's a lot of focus trying to remove those proteins in the brain. Here's another example, and you see on the right side how extensive amyloid builds up in the brain if you have Alzheimer's disease. But we've really made a breakthrough in the last decade in terms of imaging amyloid in the brain of someone who has the disease. And that's through something called PET imaging. So it used to be that you had to go to autopsy and look at the brain to find these deposits of amyloid, whereas now you can just go in a scanner and you can get an image such as the one you see on the left here, indicating that you have amyloid in the brain. And that has led to some interesting, and here, by the way, you can see a difference between someone who has amyloid, and you can see the red and the yellow, whereas the blue on top, there is no amyloid. That's from a normal person. So this has led to some very interesting findings. So this red line here represents amyloid in the brain. And you can see at the bottom, you have the age of the individual. It turns out that if you move a little bit to the right, if you have mild symptoms or if you're diagnosed with dementia and you're able to trace that person back many years, it turns out that the amyloid in the brain in that person builds up 20 to 30 years before any symptoms. 20 to 30 years before any symptoms, which leads us or brings up the opportunity to intervene before you have any symptoms if you could somehow find a biomarker or an imaging finding that would predict someone who will eventually get dementia. And that brings up an interesting question that I have for you now. So unfortunately, and we're working on this as you've heard today, diagnostics are a bit ahead of therapeutics, which means we're better at diagnosing Alzheimer's disease than we are at treating it at the moment. But we're working on it. But we are now able to, with the test, predict with some degree of certainty who will get Alzheimer's disease, especially at the stage where you have mild symptoms. So if you could have that test, we have the technology available now, but it becomes an issue of whether you would want it or not if there's no test to do something about it currently. So I'm going to bring up, so I want to remind you again, you have your white clicker. Make sure that if you have an orange button on the top, click it, make sure it's the green light. If you have a long, thin white one, click the bottom button and make sure you have a blue light. So I'm going to bring this poll up here. So I simply want you to tell me, if you could have this test, would you want it or not, if there's no treatment? And just to give you a sense of some of the answers that some people may think of, it may be no, you don't want it because there's no treatment. Whereas someone else may say, yes, I do want it because I want to plan my life ahead, I want to get my affairs in order, and it's assuring to know what we're dealing with. So on your clicker, A is no, you do not want the test, and B is yes. So I need some more. I need some more votes. So only 7. Oh, it's not working. All right, try again. Click yes or no. So A is no, B is yes. A is no, you want the test. B is yes, I want the test. Okay. So we need some more. I know there's more out there. Make sure you have a green light on your clicker. All right, so still going. So about 75, so about 2/3 or 3/4 in the room would say that they would want the test. So thank you. That's very helpful for us to know as we try to ask the province and other governments whether they can implement this testing in Canada. So I've told you about amyloid. That's something that we try to remove. I tell you about a few trials that attempt to do that, and some are more prominent than others. But there's another thing we try to do and try to reverse in Alzheimer's disease, and that has to do with sugar or glucose. So it turns out your brain loves sugar, but in someone who has Alzheimer's disease, the brain has trouble using sugar. And that's what this test here shows. Can you see what you're trying to look for here? This is sort of normal sugar uptake in the brain. This nice dark rim here. Can you see how bland that is? How weak the signal is? So that just indicates that the brain in that area is not using sugar as well. This is another way to look at it, a little bit easier. If you see blue, that means that area in the brain is not taking up sugar as well as it should. So we call them therapeutic targets. So I've told you about amyloid. I've told you about tau. I'm not going to discuss trials with tau today, but I told you about amyloid in the brain that we try to remove, and I've told you about the reduced ability of the brain to use sugar. So we call those therapeutic targets. So now you know we're going to try to do something about amyloid in the brain. We're going to try to do something about the way the brain uses sugar. So what about amyloid? Well, let's remove it. Sounds pretty easy. And there's been lots of efforts trying to do that, and there are some ongoing, very exciting efforts trying to do that. The other alternative is to try to neutralize it. So you can leave it in the brain, but whatever toxic effect it has on the brain, you can try to neutralize. What about the sugar? Well, let's give the brain something else to eat. And you can do that, as I'll show you, with certain interventions. So let's talk about amyloid first. So I have this up here. This is my version of Pac-Man. So think of these drugs as the Pac-Man in the brain trying to go and eat up amyloid deposits. I think that's the easiest way to look at it. So there were 2 recent trials. This is now about 2 years ago, very large trials in Alzheimer's disease, using this Pac-Man. We call them antibodies, but same thing. They tried to eat up the amyloid in the brain, and they didn't work. They didn't work very well. It was a big disappointment in the field. They looked at sort of middle-stage Alzheimer's disease in that group. And I'll tell you a little bit more about that in a moment. There's a new drug that tries to do this, and I mentioned this. There are several others, but I mentioned this in particular because the way it was developed is so interesting. So they took healthy, older individuals, and they asked the question, why are they healthy? Why don't they get Alzheimer's disease? And they isolated 2 of these Pac-Mans, if you will, or 2 antibodies, or 1 antibody, against amyloid, naturally occurring. So they thought that the reason why those individuals are so healthy is because they have this naturally occurring drug in their body. And then they took that and isolated it, and they made a drug out of it, and that's called, try to say this 10 times very fast. They made this drug, and now that seems to be, at least from their early studies, seem to be an interesting and promising therapy. So they showed this year, or last year rather, that over a year period, if you used this drug, you could reduce amyloid in the brain to a substantial degree, and that was associated with an improvement in memory and cognitive function. The problem, as you will see, I won't discuss it in great detail, but there are some side effects related to this drug which are being worked out now in larger trials. But of all the drugs that try to remove amyloid in the brain, this is probably the one that looks the most promising. And we have this trial at UBC. We are enrolling, as you can see in this more comprehensive review that you have in front of you, and we're recruiting patients for that study. So I want to mention one thing. Some of the trials were negative 2 years ago. And I want to mention that they were, this is sort of at where you get a diagnosis of Alzheimer's disease or dementia, and this is the stage where they were treated. Now, as you can see, the amyloid in the brain has sort of plateaued by then. It doesn't seem to change much. So we're thinking that maybe it's too late to treat here. Maybe we have to start a little bit earlier. And that's what the 2 trials that were already mentioned by Dr. Feldman earlier tries to do. What if we could identify people here where you have no symptoms, but you have a brain scan or other biomarkers suggestive of a progression to dementia later on? That seems to be the ideal stage. I liken that to a colonoscopy, for example, to prevent colon cancer. If you can catch it early enough, maybe we can prevent Alzheimer's disease. And that's what these 2 trials try to do. They're called A4 and Diane. They're a little bit different because Diane uses patients that has this familial disease, as Emily told you earlier. But it's the same principle. You take asymptomatic people with a biomarker, usually a PET scan, as I showed you, a positive on that scan. They have high risk of developing Alzheimer's. They get treated with a drug to see if we can prevent the onset of symptoms. And both of these trials are enrolling, although Diane is no longer enrolling because it has closed enrollment, but both are at UBC. What about neutralizing A-beta? So this is one of my favorite slides. I love it because it shows you an individual nerve cell in the brain. You have trillions of these, but this is one individual part of a nerve cell. And you see these little protrusions here? So those are the individual contact points between 2 neurons, 2 nerve cells. And that's basically who makes you who you are. So the more of those connections you have, the more complicated they are. And the variety of connections is really what makes us so different, and our personality is so different. So if you take one of those cells and you add amyloid, it slowly makes the nerve cell die. And that's shown here. You see these arrows. This is actually the same nerve cell, but you add amyloid, and see this protrusion here, it's gone. It's gone after you add amyloid. See this protrusion here? It's gone. So you lose connections, and that's exactly what happens. So what if you could prevent that? What if you have amyloid in the brain, which is sort of drawn here, and you can somehow prevent the interaction with nerve cell, so prevent the toxicity of A-beta? You don't have to remove it, you just have to neutralize it. And that's what we're trying to do with something called the CONNECT trial. This was actually a cancer drug. It was developed for cancer, but it's now been repurposed for Alzheimer's disease, and it attempts to do exactly that. It tries to prevent toxicity of A-beta. It doesn't remove it, but it prevents the toxicity of amyloid. We're enrolling at UBC. There's 20 trials now, 20 sites rather, across North America. So I want to show you one other clinical trial that we're doing at UBC. So I told you that you could give the brain something else to eat. So it turns out that other something else to eat is something other than sugar. So the brain loves sugar, but it also loves something called ketones. So if you don't eat for a little while, your body develops ketones naturally. If you take your favorite thing to take these days, coconut oil, you may ingest some of these fatty acids. They're called medium-chain fatty acids. They're contained in coconut oil, which is maybe why some people have reported a response to that, although we're not sure. But if you have ketones in your body, the brain can utilize ketones instead of sugar as an alternative fuel, and it may improve brain health. So at UBC, we're starting a trial called the MINT trial, where we're essentially using the active form in coconut oil under regulated circumstances, so we know exactly how much we're giving, to see if we can reverse some of this sugar dysfunction in the brains of someone with Alzheimer's disease. We're planning to enroll that in the spring. So the last thing I would say is I've shown you a few sort of a glimpse of what's going on and what you might expect in the next few years. But the last thing I would say is get involved. You know, we need to provide education to everyone here and the province, but get involved in these studies. seek information. We're here to help. And as Dr. Wellington and others have noted, we also need financial help, and we're not suggesting that you here, it's not a direct plea to you, but get the word out. We need support to do this kind of research to find a cure for Alzheimer's disease. Thank you for your attention.